the biochemistry behind the action of an RNA extraction. It's a bit like a biochemical magic trick, where we take in a series of steps, we give molecules a choice of which liquid they want to hang out with. And at the end of the day, we've got pure RNA. TRIzol, aka aka guanidium thiocyanate phenol-chloroform extraction, is a way that we can extract RNA, so separate it from everything else, such as like in a sample of cells, isolate just the RNA. And it's really useful because often you want to, for example, see if that RNA, like what genes it contains, like what mRNAs it contains, and so like what the cell was making at that time. And there's also a bunch of other uses that you'd want to isolate RNA. And with this extraction method, what we're doing, it's a liquid-liquid extraction. Taking a solution with all sorts of stuff, proteins, lipids, DNA, RNA, and we're going to make it so that only the RNA wants to hang out in this phase. And everything else is going to want to hang out down here or in this middle. So the basic idea with a liquid-liquid extraction is that you have two phases. So here we have this aqueous phase, which is all watery, and then down here we have this organic phase, which is where the phenol and chloroform and other stuff hang out. And this stuff is like where the hydrophobic stuff hangs out, so the stuff that water doesn't like. So water is going to exclude all of that, and that's all going to be here. The basic idea is that we go through different steps and we give the molecules a choice, like do you want to hang out in the aqueous phase or the organic phase, and then we can selectively get the different molecules by altering the conditions and adding different chemicals and stuff, get it so that just the RNA wants to hang out in here and then everything else wants to hang out down here. So you can see that we have these two distinct layers, and this is because these liquids are immiscible, so kind of like oil and water. So when we talk about like solubility for like solid things, we typically talk about solubility. For liquids, we can talk about miscibility. So if two liquids are completely miscible, then you can dissolve them entirely, like they'll completely mix together. But you can see that these are immiscible, so they're not mixing together. But so when you like invert the tube and mix it all up, you're giving the molecules a choice. So they're going to prefer one of the liquids over the other. And so then they'll separate based on that. And then we'll get really good separation when we like centrifuge it, so we spin it down. So that's the basic idea. And now let's go into a little more detail. Start with an overview of where we're going, and then I'll go into more detail on the various steps. So the first is homogenization. So this is basically you're breaking open the cells if you're starting from cells. So sometimes you're just starting from like a partly pure mixture or whatever, but you're gonna need to homogenize it. So basically like grind it up or shred it up or whatever to make it all just like a cellular gunk. And then you use guanidium thiocyanate and phenol. So guanidium thiocyanate it's called a chaotropes salt, and so it kind of interrupts various networks of bonding. And what this does is it's going to denature and like unfold the protein. In association with phenol. So phenol is this molecule that has this ring and this OH, and it's going to help kind of keep the protein soluble, but soluble in this unfolded state where all of the regions of the protein that were like hydrophobic, so water excluded normally, well now they're exposed. So it's going to be soluble in the phenol part, in the organic part. And if we're at a low pH, for reasons I'll show, the DNA is going to go in there too. So then what you're going to do, so you do this first and you get like this part, then you add chloroform. And so chloroform is going to make the organic solution, the organic phase, even more nonpolar. And it's going to, importantly, it's heavier. So Phenol and water, in addition to phenol being partly soluble in water, so you still have like your liquid and water and like vice versa, so you're losing some of your aqueous phase into the phenol. They have similar densities, so you're not getting like a sharp distinction between layers. But when you add chloroform, chloroform is going to make the organic phase denser, so it'll sink to the bottom. And it's going to pull the phenol in with it. So the phenol wants to go with it instead of the water. And so now you're giving the phenol kind of like a cooler option. And so it's fully miscible with the chloroform. And so it's going to form this phenol chloroform layer. Um, and this layer, so when you initially do it, it's all like foggy and cloudy, and then you spin it down, so you centrifuge it really fast, and you get this nice separation of the layers where you have this organic phase, and the aqueous phase and the interphase. So the interphase is kind of like precipitated stuff in the middle. And so your aqueous phase, that's where the RNA is going to be, as well as salts. Just why we need to do a bunch of washes to get rid of them in part. And then in the organic phase, you have like those unfolded proteins as well as the lipids. So the like cellular membrane bits and DNA, as well as some DNA in the interphase too. I'm not exactly sure. I've seen it listed both places, and so I'm guessing it's a bit of both as to where the DNA is. But because you have this separation of the RNA from the other factors, if you only care about the RNA, then all you need to do is take this aqueous phase and go from there. There are also, though, however, like the reason it's called like TRIzol or Tri, so I think TRIzol is the one brand's name is TRIzol. I think it's in Vitrogen, and then Sigma is like Tri. There's also like make it yourself versions. But the basic idea is like with the try for like three things. So the try for like three things. So the RNA, DNA, protein, you can actually, after you take out the RNA, you can then further like back purify and back extract and get the DNA and DNA out, and also like the proteins, you can try to like resolubilize them and stuff. But anyway, what we're interested here is the RNA. And so we're going to take this aqueous phase and put it in a new tube. And now we need to precipitate out the RNA. So we use this extraction to extract it, but now we need to like purify it. So it's in this liquid now. It's separated from most of the other stuff, but it's still with a bunch of salts and stuff. And there might be some contamination still from the proteins and lipids and DNA and stuff. And so we're going to need to precipitate the RNA. And so now we need to get it to come out of solution. So we changed what it wanted to be dissolved in by giving it this other option and making the original option less desirable for the other molecules. So now we need to get the RNA out of solution, so out of any solution. And so we do this by adding isopropanol. So isopropanol is going to help. So it's like it's more nonpolar than the water, but less so than the like the organic phase that you had before. So you're giving it kind of like an intermediate option. Or I guess you're not really giving it an option. You're lowering the like polarity of the solvent in general, because the isopropanol is going to be like miscible with the water. So now you have this solution of isopropanol in water. And the isopropanol is kind of like going to make the water less attractive for the RNA. And you're also adding, you have all those salts in there. And when you have the water-based solvent, the salts kind of can hang out with the water, and they're all cool. It's kind of hiding the salts, so hiding the like positively charged ions and stuff in there that could otherwise be clinging on to the RNA's negatively charged backbone. So when you add the isopropanol, what you're doing is you're like lowering that, we'll talk about this more, but you're lowering the dielectric constant, and you're kind of like making that like fog, but like making it molecularly less foggy in terms of charges being able to see one another. And so then those negative and positive charges can see one another. And now you have the salts are going to neutralize the backbone of the RNA. And so that makes them non-charged. And when they're not charged, now they're not soluble. And so they're going to precipitate out. And so you don't really give them a different liquid option. All their option is that water isopropanol solution. And so they're going to precipitate out. And then you can remove all that other liquid. And so now you have a pellet of RNA. But it's probably not that pure yet. So what you're going to do is you're going to wash it. So you had a solution of ethanol. And so ethanol, now we're going back more towards water, but not as much. So this more water-like than isopropanol, but not water-like enough that the RNA will dissolve in it. But those salts will dissolve in it. And so they're going to, you can wash them off and then remove the liquid, so you're removing all those salts, and then you can resolubilize it. So now you add your water and dissolve it again, and now it's all by itself, and then you air dry it to evaporate any of the lingering ethanol. Okay, so that's the overview of where we're going, and now let's look in more detail. So before we start, some key things about working with RNA. And so RNA, you need to be really careful to avoid like contaminants and things. So there are these things called Rnases, which are basically like out to get you, and out to get your RNA. And so they're kind of like RNA scissors, and they're basically everywhere because a lot of viruses are like RNA based, and so all of like our cells and the bacteria and everything, they make these like RNases to try to prevent like RNA-based viruses from like messing with them. And they even survive in autoclave. It's pretty amazing. But we can use this molecule called DEPC to try to inhibit these RNases. So RNAs, you don't need to know this, but its activity like depends on this action of this cysteine residue in the active site, and DEPC is going to kind of like stick to that part and activate it, and then you autoclave it, and that inactivates the DEPC, so you don't mess up anything in the future. But this is also the reason, one of the main reasons why it's important that you have this guanidium thiocyanate phenol step where you're actually like denaturing the proteins, because you can help denature any RNases that might be in there before you start this whole thing. And so also throughout the process though, because remember the DEPC, even that like it's you've inactivated the DEPC when you do the autoclaving. And along the way here, you could still have RNase contamination. And so typically, when we're working with RNA, we'll use like RNA zap, this is like thing, and wipe down with like ethanol and stuff like our pipettes and everything, and be really careful when not to introduce contamination, because the RNAs can like kill things. And also, if you're doing this in turn, like because you want to do like qPCR or some other thing, where you're going to want to see what RNA is in there, you don't want to accidentally introduce foreign RNA. And so it goes both ways depending on your what you're looking to do. So throughout this biochemical magic trick, what we're really doing is we're exploring molecules' solubility in other molecules and their solubility in general. And so basically, molecules, in order to be soluble, something has to have like every piece like has its complete water code. So for our RNA to be soluble in like water, that RNA, each piece of RNA is like this full water coat. But water also really likes to bind to itself. And so in order for something to dissolve in water, the water has to like it enough to bond to it. And then the thing that you're dissolving has to like the water enough to bind to it. So if the solute molecules, so we have like a thing that's being dissolved, we call it a solute. So if they like each other more than the solvent, they're clumped together. And then if they like the solvent and the solvent likes them, then they'll each get their own liquid coat and we call it soluble. And so here we're talking about solids and liquids, but we can also think about that happening in liquids and liquids. And so the phenol and chloroform, they don't like the water, and so they're going to separate out and not be dissolved liquids. And so the reason why molecules like do or don't like each other, it comes down a lot to polarity and partial charges. So molecules are made up of atoms, and these atoms are made up of smaller parts, and so you have protons, which are positively charged, and neutrons, and those hang out at the center in the central core called a nucleus. And then the electrons are these negatively charged parts that whiz around them. And so molecules are going to share pairs of electrons to form bonds, but they don't always share fairly. And so we can get this thing called polarization, where even though a molecule might be neutral overall, it has charged parts because they're not sharing those negatively charged electrons fairly. So the like in water, the hydrogens are going to be partly positively charged, and the oxygens are going to be partly negatively charged because the oxygen is going to be pulling tightly. It's hogging those electrons more. And so this, you can get polarity, and because opposite charges attract one another, then things with partial charges will be attracted to things with the opposite partial charges, and things with full charges will be, and that sort of thing. So we call ions molecules with full charges. And so salt is actually a combination of like a positively charged ion and a negatively charged ion. But we could also have parts of molecules where they're polar, so they have parts that are charged and parts that aren't charged. And so basically, when we talk about like a polar solvent and that sort of thing, and like polar molecules, sometimes like with something that likes polar molecules will dissolve in, like water. So water is polar. It has it's highly polar, so it likes to form these water networks. So it's also the reason why it's forming those networks, remember, is because of those partial charges. So it's also going to be really soluble for things that are fully charged, like ions. One of the places that you will find full charges is in the backbone of DNA and RNA. So DNA and RNA are made up of these nucleotide letters, and so they have this negatively charged phosphate backbone. And then these bases, which is what they use to form pairs to each other. But the key thing in terms of this is like these backbone phosphates make them really soluble in water, because remember, water likes charged things. And something that's nonpolar, it doesn't have like any charge or even any separation of charge, or sometimes we call things nonpolar, but they're really like slightly polar, but not enough to really matter that much. And so those are the things that are going to go into that organic phase. But RNA and DNA, because they have this phosphate, you'd think that both would want to be in the aqueous phase. And if you were to do this at a higher pH, you would see that. So pH is a measure of proton availability. So protons are H+, and what happens is that at a low pH, these nitrogen on adenine and cytosine, so two of the bases, these can actually get protonated. So a proton get picked up from the acidic solution, and that makes this positively charged, which is going to partly cancel out the negative charge on that backbone and make them less negatively charged, so less charged, and that charge was the main reason that they were staying soluble in the water. So that can also happen to the RNA bases, but the RNA, it has this extra OH group. so that deoxyribonucleotides and ribonucleotides. So one of the main differences between DNA and RNA is that they have this different sugar in their base. And so the RNA has this extra OH group. And remember, OH is, an O is really electronegative, so it's going to pull on those electrons and make this more polar. So it's going to be able to stay soluble even at a lower pH. But the DNA will become more neutral. And so it's going to then precipitate out and be in that interphase, or it'll be soluble in that organic phase, so because it's more neutral. When you have those protonation of the bases, you're messing up the way that they interact with one another. So the DNA likes to be in these double strands. And those double strands are hiding the most hydrophobic parts of the DNA, like in the center. So the negatively charged backbone's on the outside, but then you have these bases in the center. And so normally these bases are hanging out together, but when you protonate them, you're messing up the ability to base pair with one another. And so this is going to make the strands denature and come apart. When they come apart, now they're exposing all those hydrophobic regions that they were trying to hide. And so those are going to like the organic solvent more. So you'll get the DNA going into that organic phase, and also it can like precipitate and hang out in that interphase. You're neutralizing the charge and make it so nothing really likes it. The RNA at that point is still saying soluble, but it won't be soluble when you switch it to the isopropanol. And so isopropanol is commonly called rubbing alcohol or isopropyl alcohol. And you can see that it has these these like 3 carbohydrates and just that one OH, and no charge or anything. And so it has this OH, so it has this polar part, but this is giving it a lot of nonpolar character because these carbons and hydrogens share fairly, their electrons pretty fairly. So this isn't going to be blah. So this is kind of like a blah nonpolar part. And so this is going to make the solution less desirable to the RNA. Dielectric constant has to do definitely with the ability of a solution to neutralize a charge, or to keep 2 charged molecules from finding each other. And so the higher the dielectric constant, the more polar the solution, the more these molecules can orient themselves in a way that will arrange around the charged things in order to keep the charged things from finding each other. At least that's my explanation of it, and hopefully that's completely accurate. But the key thing is that if you have something that's highly polar, like water, it's going to have a high dielectric constant. And this means that if you have charged molecules in there, the water molecules are going to hang out around those charged things. Because remember, water, it's highly polar because it has these partially charged regions. And so partially charged regions are going to like fully charged regions. So they're going to be attracted to these salts and these ions, and they're going to coat these ions. And then they're also going to coat the RNA. So you have all this water. It's going to be hanging out around both the RNA and around the, because remember the RNA has that backbone, and then it's going to be hanging out around the salts. And our goal is to get the salts to hang out with the RNA, because right now the water is hiding them both, because we have that high dielectric constant. So the water's kind of hanging out with everything. So we want to kind of reduce the amount of water and with something that is less polar. And so when we add isopropanol, we're lowering the dielectric constant. So it has a lower dielectric constant. When you mix the two, you get the lower dielectric constant of the overall solution. And so basically what this means is that now you have fewer water molecules available to shield those charges, and your solution overall has a less ability to hide all those charges from one another. And this means that now you can have your RNA get neutralized. So those those cations can find the RNA. And when they find the RNA, they're going to neutralize its backbone, just like we saw with the DNA. The DNA is going to get, the RNA is going to get neutralized now. And then we can, it'll precipitate out. Add the isopropanol, you're going to help the salts find the RNA backbone. When they do that, they're going to hide the charge of the backbone. The RNA becomes uncharged. Now it's going to precipitate out. And when it precipitates out, now you can get rid of all the other stuff that was around, like the phenol and the salt. So to get rid of the salts, what you're going to use is an ethanol solution. So ethanol, that's just an ethyl and an OH. So this is more, this is even more water-like, but it's not water-like enough to dissolve the RNA, because you have these big things of RNA that are all kind of clumped up. So you resuspend the pellet, so you kind of like pipette it up and down in this ethanol solution, and then you centrifuge it again, so you spin it down, and this is going to remove the ethanol, so the ethanol is going to dissolve. This is going to remove the salts, so the salts are going to dissolve in this ethanol solution. So it's not pure ethanol, it's about like 75%. So it's good enough for the salts, but not for the RNA. And so you do this a couple of times to get rid of any lingering salts. And then you can remove that liquid and air dry it. So don't vacuum dry it. Don't let it like over dry, or it's going to be hard to dissolve. And so you also might want to dissolve it in like heat up the solution while you're dissolving it, and you can dissolve it all by itself. So often when you do this dissolving, what you're going to do is you're going to do it in a depth sea treated water. At the end of your purification, you're going to want to check and see how pure it was. And so you often do this with like spectroscopy. So often like this nano drop. Basically the idea is that you shine light through and the see how much light gets absorbed. And because like different molecules absorb different wavelengths, then you can get an idea about how pure the thing is. And so there's three values that you want to look for: 230, 260, and 280. So 280, that's where we have like protein predominates. And then, so this can tell you, you look at the ratio. And so for like pure RNA, you want about a 260 to 280 of around 2 and a 260 to 230 of greater than 1.8. And so 260 over 230 is going to tell you about like contamination from phenol and salts. And 260 over 280 is going to tell you about contamination from proteins. And so this is one of the reasons why it's important to do that, like those ethanol steps, to really get rid of these salts and phenol. So I hope that helps you, and I hope that you have a successful extraction. So just a last note. So this is just one of many ways for RNA extraction. It's based on this method from like 1986. See, if you make a methods paper that gets really popular, you get ton of citations. But anyway, so this is where it came from. There are other methods as well. Like there's a lot of easier methods like kit-based methods, which use like spin columns, kind of like you might have seen with like mini preps or PCR purifications. But so those are easier, but they tend to give you a lower yield and potentially less product. Depends on like how you do things and everything. But that's just what I've read, and it's also less suited for various purposes. But yeah, so definitely look into it, and then you can find like all the information. You can make all these reagents yourself, and do this sort of thing. And then one last note I wanted to mention was that when you do the precipitation, it can be really hard to see the RNA. So you're gonna want to, you can like try marking the tube, and it's when you have the centrifuge like this, your pellet's gonna be like on the outside of right here. So just be really careful, because it might be hard to see, especially if you don't have that much RNA. Okay, well, hope that helps.